Dear students, myself Dr. Dipti Srivastav, Associate Professor, Government Dungar College, Bikaner. Today we will discuss on physiology of excretion in insects. The secretory system eliminates nitrogenous waste products, excess of salts and water, amino acids, pigments, and maintains their concentration. Contents: mechanism of excretion, urine and excretory end products, storage of excretory products, control of excretion. Mechanism of excretion: main waste product is uric acid. Tubes branching off intestinal tract called Malpighian tubules. Ions and uric acid are actively transported into lumen of these tubules. Water follows through osmosis and finally uric acid excreted. Terrestrial insects are the euryotelic animals like reptiles and birds, and their principal excretory product is uric acid, while the aquatic insects are often uricotelic, excreting mainly ammonia. In the hemolymph, uric acid combines with sodium and potassium carbonates or bicarbonates to form sodium and potassium urate respectively. Sodium and potassium urate salts are highly soluble in water and are easily absorbed by the Malpighian tubules as they can cross the cell membrane in solution. In insects like Rhodnias, the tubules are differentiated into two parts, proximal having brush border and distal with honeycomb border. An aqueous solution of urates is always absorbed by the distal part. The urates gradually accumulate into the lumen of tubules at distal part and slowly reach the proximal part. During their transport from the distal to proximal part, uric acid is precipitated. In alkaline pH, enzyme carbonic anhydrase splits urates to degenerate the bicarbonate and uric acid. The carbonic anhydrase is secreted by epithelial cells of the Malpighian tubules, and subsequently water and carbonates or bicarbonates are reabsorbed into the blood so that only uric acid is left into the lumen of tubules. The water and carbonates combined with the uric acid form urates, which can be excreted in the similar way in a next excretory cycle. The semi-solid uric acid particles move into the lumen of hindgut. Inside the hindgut, again the precipitation of uric acid takes place through reabsorption of water and new other metabolites by iliac or rectal wall into the hemolymph. In Carouseuses and other insects in which there is no differentiation of Malpighian tubules into distal and proximal segments, the entire tubule functions as a distal part absorbing only urate salts, while conversion into uric acid occurs in the rectum. In dipteral larvae, rapid conversion of urates into uric acid takes place through the Malpighian tubules, suggesting functional differentiation at the cellular level rather than regional one. Continuous release of large quantity of water from the Malpighian tubules to the rectum drives uric acid along it, and ultimately it is excreted through feces via the anus. It is now well established that potassium is actively transported into the Malpighian tubule while sodium by simple diffusion. Various metabolites having small molecules pass through the wall of tubules under the influence of a concentration gradient. The excretory mechanism in true sense therefore involves filtration of the fluid of hemolymph, excluding the proteins which cannot penetrate through the membrane due to their large molecular size. Hence, it is a principal device of osmoregulation. By eliminating excess of salts or water or by converging them depending upon the physiological conditions, it maintains the ionic and water balance of the hemolymph. So, in during the excretion, the Malpighian tubules are bathed in the insect's blood, but since they are not rigid, it is impossible for any hydrostatic pressure to be developed across their walls, such as could bring about filtration. The primary urine is formed by a process of secretion in the following way. Potassium ions are actively transported from the blood into the cavity of the tubule and are necessarily followed by negatively charged ions so as to maintain electroneutrality. In turn, water follows the ions, probably by osmosis, and various other substances, sugars, amino acids, and urate ions also enter the primary urine by diffusion from the blood. Sugars and most amino acids are also passively filtered from the hemolymph via junctions between the tubule cells, whereas amino acids and non-metabolizables and toxic organic compounds are actively transported into the tubule lumen. Sugar is reabsorbed from the lumen and returned to the hemolymph. The continuous secretory activity of each Malpighian tubule leads to a flow of primary urine from its lumen towards and into the gut. In the rectum, the urine is modified by removal of solutes and water to maintain fluid and ionic homeostasis of the body. The primary urine, together with soluble products of digestion and insoluble indigestible matter from the midgut, then passes to the rectum. There, or in some insects at earlier stage, the urine is acidified and the soluble urate is thereby converted to insoluble uric acid, which comes out of solution. Water is then reabsorbed together with the soluble products of digestion and other useful substances, including the bulk of the ions that entered the primary urine. In insects that live in dry surroundings, the rectum has remarkable powers of reabsorption, its contents finally being voided as hard, dry pellets containing solid uric acid. Here we can see the lumen of rectum, hemolymph, transport of water molecules through simple osmosis and Malpighian tubule cells water enters through hemolymph into Malpighian tubules and then lumen of rectum transcellular route of uric acid, alkaloids and salts, paracellular route through gap between two cells, two epithelial cells of rectum. Through this route, sugars and amino acids are transported into lumen of rectum. Sodium ions, potassium ions and chloride ions actively transported into the Malpighian tubule cell and then lumen of rectum. In this diagram, we can see Malpighian tubule of Rodnius in A diagram, and in B we can see Malpighian tubule of Caroseus. As we know that in Rodnius the Malpighian tubule is divided into two parts, proximal having brush border and distal part with honeycomb border. The aqueous solution of urate is always absorbed by distal part. And in Caraseus there is no differentiation of this type of parts. There is no differentiation into distal and proximal segments or parts. The entire tubule function as a distal part absorbing only urate salts, and conversion into uric acid occurs in the rectum. So, continuous release of large quantity of water from Malpighian tubule to the rectum drives uric acid along it, and ultimately it is excreted through feces via the anus. Cryptonephry. The distal ends of the Malpighian tubules are held in contact with the rectal wall by the perinephric membrane, which is concerned either with efficient dehydration of feces before their elimination or ionic regulation. Example: adult Coleoptera, larval Lepidoptera and larval Symphyta. As we know that in cryptonephrons the distal ends of Malpighian tubules are held in contact with the rectal wall by the perinephric membrane of rectum. So here we can see that the direct contact of Malpighian tubule with rectum. Urine and excretory and products. The physical and chemical properties of urine vary among insects according to their food and environment. In fluid feeders, that is plant sap or blood, urine appears as a crystal clear fluid. In aquatic insects, urine is copious and clear. The percentage of water is often higher. In the terrestrial and marine insects, urine is excreted in dry pellets or semi-solid form. In stored grain pests, it may occur in bone dry powder form. Pediculus discharges a moist white pellet. The urine charge changes from alkaline to acid as it moves from Malpighian tubules to the gut. Due to gradual fall from pH 7.2 to 7.8 to 6.0 to 6.66 respectively. The The chemical composition of urine also varies in accordance with the nature of food and physiological adaptation of the insects. It contains water and salts. The salts include bicarbonates, chlorides of sodium and potassium, phosphates of magnesium and calcium, sulfates derived from metabolism of uh, cysteine and methionine amino acids of protein molecules, nitrogen in the form of uric acid, ammonia, urea and amino acids, and pigments and other trace constituents. Nitrogenous and products. These are uric acid, ammonia, urea, amino acids, pigments and other trace constituents. Nitrogenous end products. First, uric acid. It is the nitrogenous end product of excretion in terrestrial insects in which water conservation is very essential. It requires little quantity of water for its discharge from the excretory system. In pupae also the end excretory product is uric acid. About 28% to 85.8% of total excreta is represented by the uric acid. The uric acid is scarcity of water is excreted in the form of crystalline spheres or spherical granules of 1 to 60 diameter. The urates may be associated with the free uric acid, which represents ammonium urate, calcium urate or sodium and potassium urates. The uric acid is derived from metabolism of purines, adenine and guanine, and or from the proteins by utilizing glycine, glutamine and aspartate as substrates. Here we can see the formation of uric acid from the metabolism of purines, that is adenine and guanine. Guanine monophosphate is converted into guanosine in the presence of nucleotidase enzyme. Guanosine converted into guanine in the presence of nucleoside phosphatase. phosphorylase enzyme. Then in the presence of guanase, guanine is converted into xanthin. Xanthin is now converted into uric acid in the presence of xanthin oxidase. Similarly, adenine monophosphate converted into adenosine. Adenosine is converted into inosine in the presence of adenosine deaminase. Inosine now converted into hypoxanthine. Hypoxanthine form xanthin in the presence of xanthin oxidase enzyme. Xanthin forms uric acid in the presence of xanthin oxidase. So, in the metabolism of purines, adenine and guanine, ultimately uric acid is formed. Next excretory product is ammonia. In the larvae of Calliphora and Lucilia, which are meat feeders, excrete large quantity of ammonia as the excretory end product. It is also excreted by the aquatic insects as the main nitrogenous component of the urine. It is a very toxic substance and its elimination requires a large quantity of water in the urine. In some terrestrial insects also, sometimes it occurs in a very small quantity in the dried excreta, example Melanoplus. 0.6 to 0.7 milligram of ammonia is excreted along every 10.6 to 15.6 milligram of uric acid. Next excretory product is urea. The insect urine often contains a small quantity of urea, that is 0.3 to 0.4% of dried excreta in Melanoplus and about 10% of total nitrogenous waste products in the mosquitoes. Here we can see the formation of urea through citrulline. Citrulline forms arginine and when arginine converted into ornithine, urea is formed. Ornithine ultimately converted into proline. In this figure, we can see the formation of the excretory products in insects. Uric acid converted into allantoin in the presence of uricase. Allantoin converted into allantoic acid in the presence of allantoinase enzyme. Then allantoic acid converted into urea in the presence of allantoicase enzyme. Urea plus glycoxylic acid forms ammonia in the presence of urease enzyme. Storage excretion. The excretory waste materials are retained within the body in different sites. Uric acid is stored as urates in the cells of fat body, example American cockroach. Uric acid is stored in the body wall giving white color, example red cotton bug. Uric acid is stored in the male accessory glands to produce the outer spermatophora which is excreted during copulation. Uric acid is stored in the wing scales giving white color. Example parrot butterflies. Waste products of pupal metabolism is stored and released during emergence. Control of excretion. The activity of the excretory system in insects is under hormonal control. This has been most clearly demonstrated in the case of Rhodnius, a blood-sucking bug. Immediately after the ingestion of a blood meal, there is a rapid flow of urine whereby most of the water taken in with the blood meal is eliminated. The distension of the body after ingestion is the stimulus that causes certain cells in the central nervous system to release a hormone that acts upon the Malpighian tubules to promote a brisk flow of primary urine.